So my name is Graham Hardie and I'm the Professor of Cellular Signalling at the College of Life Sciences at the University of Dundee. And I study cell signalling, which is what I'm Professor of Cell Signalling, but particularly a signalling pathway which is called AMP activated protein kinase, or AMPK for short.
Okay.
I suppose what I would do is, as you know, all living things are made out of fundamental units called cells, which are just sort of usually too small to be seen with the naked eye, you need a microscope. But every cell in the body has its own power supply, which is rather analogous to the rechargeable battery you have in a mobile phone. And if I could use them and extend that analogy, what AMPK does is it monitors the energy state of the cell. So it's a bit like the system in your mobile phone that monitors the state of the rechargeable battery. And if it detects that the battery is running flat, in other words the cell's running short of energy, it actually tries to correct that. And what it does is briefly what it does is it switches on so-called catabolic pathways which sort of recharge the battery. And that's the pathways in which you take up glucose and fatty acids and you burn them off to generate energy. But at the same time it switches off pathways which consume energy, and that includes cell growth and cell division, so it tends to inhibit cell growth and Right, well, not a lot. You're talking about the late 1970s when I first started working on this, so a long time ago. It was known that there was a protein kinase. Protein kinase is a protein that modifies other proteins by phosphorylation. And it was known that there was a protein kinase that modified a key enzyme of the pathway of fatty acid synthesis, which is called acetylcocarboxylase. But at that time it was thought that that's all it did, and we had no idea what its real physiological function was. So over the years what we've shown is that it actually not only regulates that enzyme, it regulates many enzymes and proteins. In fact there's about 40 or 50 known now. And we also developed the idea that it is an energy sensor, and that's what its physiological role is. Right, well there's no doubt about that, because it happened in late 2002, and the way that AMPK is activated when the cell is short of energy requires it to itself be phosphorylated, modified by another protein kinase, and we spent several years trying to identify this other protein kinase, and it was very difficult and challenging. And then one afternoon in 2002 we got the results of some experiments which suggested that it might be another protein kinase called LKB1. And what was really exciting about this was that the person who could help me with this was just down the corridor, because my colleague Dario Lessi was working with LKB1. It was known to be a tumour suppressor, in other words a protein kinase that was involved in cancer, but no one knew what it did, what its downstream targets were. And this was one Thursday afternoon, and by lunchtime of the next day we'd done the first experiment, and it was obvious that we'd got the right answer. So that was really exciting, because for the first time it introduced a link between AMPK and cancer, which wasn't there before. Right, well, it's most commonly discussed in the context of type 2 diabetes, which is the disorder which it's a form of diabetes which is most common in older people, and it's associated with being obese, strongly associated with obesity, and also to some extent with not taking enough exercise. And it's not so much that AMPK is damaged in that disorder, but what happens is that if someone is eating too much, they're storing too many nutrients in cells, and that actually keeps AMPK in a sort of inactive state, because it's actually activated by starvation, by energy lack, by starvation. And of course AMPK is also very important in muscle during exercise, so it's activated during exercise. So if you don't take much exercise you won't get much activation of AMPK. And so it seems to be at least partly caused by the fact that not so much that AMPK is damaged, but it's just that it's under those conditions it's rather inactive, and that leads to some of the consequences of type 2 diabetes. turns out that, in fact, one of the drugs which is most commonly used to treat type 2 diabetes, which is called metformin, which was introduced in the 1950s, it's actually interesting because it's based from an old herbal medicine, but no one knew how it worked.
Right.
And it turns out that it seems to work primarily, maybe not entirely, but primarily by activating the AMP kinase. And so now that drug's already out there, but there are, and it's prescribed to more than 100 million people worldwide. So it's a really big thing. And it's a cheap and effective drug, but there are a few problems with it. The major side effect it has is it causes gastrointestinal disturbance, and that means that some patients can't tolerate it, because they simply can't take the drug, and it seems likely that the gastrointestinal side effects are not mediated by AMPK. So if we could develop a drug which activated AMPK more directly, because we know how metformin works and it's rather indirect, it's possible that it would be just as effective, if not better, than metformin, but would avoid some of the side effects of metformin. So many pharmaceutical companies are now working on this idea, and it's very likely that some of these new AMP-acting drugs will actually go into clinical trials quite soon. Well, the other exciting area has been the role of AMPK in cancer, and obviously this started with our finding that LKB1 was the upstream kinase, and that before we had no idea really that it had anything to do with cancer. But now it appears that AMPK, LKB1 is a so-called tumour suppressor. It's a protein that tends to suppress cancer, but when it's lost through mutations, that causes an increase in the incidence of cancer. And it seems likely that that's mediated by, at least partly by activation of AMPK. Another rather interesting area is that we showed a couple of years ago that AMPK was activated by salicylate, which is the natural product from which aspirin was derived. Now, it's been known for a few years that aspirin provides, regular aspirin use seems to provide some protection against the development of cancer, but no one knows what the mechanism of that is.
Right, okay.
So our recent work with salicylate and AMPK suggests a possible mechanism, because AMPK is known to have this sort of repressive effect on cancer. Perhaps at least some of the effects of aspirin might be mediated by AMPK. And then there are some problems associated with taking aspirin regularly. That also has side effects. So it's possible if we could again find a different way of administering salicylate, it might have this anti-cancer effect while avoiding some of the side effects, which again mean that some people simply can't tolerate regular aspirin use.